Hi everyone, my name is Rousseau. I'm a postdoctoral researcher at IBM, and today my talk is entitled Defective Interfering Particles, a competition-based therapy against viruses. So first, what are viral defective interfering particles, or DI particles? They are viral deletion mutants, so if you have the genome of a wild-type virus, and a part of the genome is deleted, then you may obtain a DI genome. So the first property is that it is defective, as it is not able to produce some essential elements to complete its cycle, and this is because of the genes it is lacking. And the second property is that it is interfering, because when there is co-infection of a cell by the wild type and the DI genomes, there is competition for cellular resources, and most importantly, the DI can steal its lacking elements from the wild type. So the DI acts as a parasite of the wild type virus. And DI particles have been observed in nature, for example, in influenza A, dengue, Ebola. So we can see that the DI hinders the wild type by interfering, and we would like to use DI particles as a therapeutic agent against wild type infection. So for this, we started by engineering a DI particle for poliovirus that is lacking genes encoding for capsid proteins. So you can see here a representation of the wild type genome, and the P1 region is the one encoding for the capsid proteins. So you can see that in the DI genome, most of the P1 region was deleted. So as a consequence, here are the key steps of the within-cell viral cycle for the wild-type virus and the DI particle. So first, when a wild-type virus infects a cell, its genome will get replicated, it will produce new capsid proteins, and the newly produced genomes will get encapsulated to produce new virions that are going to be released at cell lysis. And now, if a DI particle infects the same cell, the first thing is that it will replicate faster because its genome is shorter. And then, as I told you, it is lacking the genes encoding for capsid proteins, so it is going to steal the capsid proteins from the wild type to produce its own new virions. Now, before moving on, an important parameter is the burst size that we are going to see throughout this talk. So the burst size is the number of virions that are released at cell lysis. So all of this context brings us to my research question, which is to identify the critical parameters for the optimal design of an efficient DI particle. So for this, we developed a multi-scale approach, starting at the cell level, where we studied the competition between the wild type virus and the DI particle within the cell. And we did this thanks to experiments and to mathematical modeling. And we could show that there is competition for cellular resources that are needed for replication, as well as, obviously, for the capsid proteins that are produced by the wild type. Thanks to this study at this level, we could estimate from experiments the wild type and the DI burst sizes, which are again the number of virions released at cell lysis. And these burst sizes serve as input parameters for the next level, which is the tissue level, where we study the spread of the wild type and the DI at the cell population level. And then the next level is the host level, where we developed a multi-organ model. So for this presentation, I am going to focus on the tissue level. So for this, we developed an intercellular model that is based on ordinary differential equation. You can see on this slide a flow diagram of the model that is composed of a number of state variables. We have susceptible cells, we have cells that are singly infected either by the wild type or by the DI, cells that are dually infected, and then we have wild type and DI free virions, free meaning that they circulate outside of the cells and can infect new cells. Then we have a number of parameters for the birth and death of cells and the clearance of the free virions. For the primary and secondary infections with an infectivity factor K, a parameter that is representative of the infectivity of the Y-type variance, and then another one representative of the infectivity of the DI variance, which is mu. And finally, we have parameters for the release of variance at cell lysis. So we have the Y-type burst size from single infected cells, and then the Y-type and DI burst sizes from dually infected cells that are C and Rho. So you can see that this model contains a large number of parameters. So the first thing we did was to conduct a sensitivity analysis, which allows us to assess the relative importance of each parameter on a variable of interest, and in our case, we chose the proportion of Y-type free variance in the population after 10 weeks of infection. So this bar plot represents the results of the sensitivity analysis, and it represents the percentage of variance of the variable of interest that is explained by each parameter. So the larger the percentage of variance explained, and the most important or the most influential is the parameter on the variable of interest. So you can see that the three most important parameters that I highlighted in red on the bar plot are actually very interestingly directly related to the DI. So we have again Rho and C, which are the relative DI and Y-type burst sizes from dually infected cells. So they are representative of the competitivity of the DI, and then mu, which is representative of the infectivity of the DI particles. I'm not going to detail the next parameters for a matter of time, and they are also less important. So now let us focus on two of the most important parameters. So on this heat map, you can see again our variable of interest, the proportion of Y-type free variance, as a function of the Y-type and the DI burst sizes from dually infected cells. And you can see that the blue region of the heat map represents low Y-type viral load, which means that the DI is efficient at inhibiting the Y-type. And this is achieved for large Y-type and DI burst sizes. So it can seem counterintuitive to have to increase the Y-type burst size in order to decrease the Y-type viral load. But if you look at the flow diagram of the model, in order for the DI to play its parasitic role, it needs to co-infect a cell with the Y-type. And in order to do so, it's a long way because you need first primary infection and then secondary infection. So in order to increase the probability of co-infection of the cell, one way is to increase the number of free variants. And in order to do so, you can increase the burst sizes of both the Y-type and the DI. Then the black cross on the graphic represents the experimental estimations from the lab of the Y-type and the DI burst sizes. So you can see that the model predicts that our current engineered DI is already pretty efficient at inhibiting the Y-type. So second, we conducted an invasibility analysis, and this allows us to assess the dynamic stability of the DI when introduced after Y-type infection. So in other words, it is mimicking the efficiency of the DI used as a treatment against Y-type infection. So for this, we start with a DI-free equilibrium, so Y-type on infection. The system is pretty simple, and we can obtain easily the steady states. And then we introduce a small amount of DI-free variance, and we determine whether the DI-associated variables will expand or contract in time thanks to the analysis of the characteristic polynomial. So here is the result of this invasibility analysis, which represents the DI stability as a function of the Y-type and the DI burst sizes from dually infected cells. So first, the gray region of the graphic represents cases where the DI is predicted to go extinct after introduction. And this is the case when the Y-type or the DI relative burst size is too small. And on the opposite, the white region represents cases where the DI is predicted to be maintained. And this is the case when the Y-type and the DI relative burst sizes are large enough. So it is a similar result as the one of the sensitivity analysis. And again, the black cross represents the same experimental estimation of the Y-type and the DI burst sizes. And interestingly, we can see that the model predicts that our current DI is going to be maintained if it is introduced as a treatment. So in conclusions, we can see that the most important parameters for the Y-type viral load at the tissue level are related to the DI construct, so to its infectivity, and to its competitivity within cell. And then for the optimal design of the DI particle, we would need to build a moderately competitive DI within cell that will maximize both the Y-type and the DI burst sizes. And we obtained these results both for an initial co-infection with the Y-type and the DI. So this is the first sensitivity analysis that I presented to you. And we also obtained these results in the case of a DI treatment after Y-type infection. So this is the second invasibility analysis. So as perspectives, we would like to study the potential of vesicles that were shown to carry hundreds of virions in poliovirus. So it means that these vesicles represent a potential to increase the probability of cell co-infection by the Y-type and the DI, and thus they could increase the efficiency of the DI. And then we would like to study the impact of the immune response on the dynamics of the Y-type and the DI. I would like to acknowledge the people that have been working with and who contributed to this work. Simone Bianco is my supervisor at IBM, and then we have been working with researchers at UCSF in rural Montana. With this, I would like to thank you for your attention and welcome any questions.
Yeah, do you have any sense of, you're co-infecting these things, the reversion rate to like a functional particle? I mean, viruses are the masters of molecular evolution.
Yeah, we didn't do any experiment yet to study that, but we are concerned about the safety of the DI particle, and in the future we will make sure that we will introduce some elements in the DI particle to make sure that if there is recombination, the newly produced genome will not be viable. Yes, but we don't know the rate of recombination. Thank you.
I have a question. Does the burst size change over time with these co-infections? Like, would you expect that cross to move over time in any of the directions?
Yeah, I guess if there are mutations that come in the genome, the burst size can change. And also, I did some simulations at the within cell level, and I could see that if there is a delay between Y-type and DI infection of a cell, then the burst sizes are going to change significantly.
So if you add in that immune system stuff in the future, do you think it's going to start interfering with the integrated particles because of eliminating also like the radiant
it's difficult to predict what is going to happen. But we know that the DI particles have been shown in several papers to induce more immune response than the Y-type virus. They induce more interferon 1. So yeah, we don't know what is the role of this, and we would like to do simulations to see what is happening. But I think it will decrease the viral load, and it may be not so good for the DI particle to be used as a therapeutic agent. I think another 14 minutes. Thank you.